A warning. This episode contains references to violent crimes. Please listen with care.
A LiSTNR production.
DNA is increasingly being used to solve crimes. And we're now starting to learn more about the limitations of DNA and how it affects prosecutions and defence in criminal trials. Jae Gerhard is Principal Forensic Scientist at Independent Forensic Services. Starting out as a forensic scientist at the Australian Federal Police in 2002, Jae is an expert in body fluid detection and confirmation, bloodstain pattern analysis, and DNA analysis and interpretation. Jae is going to take us through the process of DNA collection, how accurate it is, and how it's processed. And DNA's become so sensitive, it's become less useful as evidence. So, what comes next? And in the second half of this episode, Jae tells us about Stain 91, a blood stain that played a part in helping to solve one of Australia's most renowned murder investigations. My name's Kathryn Fox. I'm a former GP, crime author, and screenwriter. I'm enthralled by forensics and have spent thousands of hours researching for books and screenplays. So I thought, why not turn my research into a podcast? Every week, you'll be joining me in discovering how forensic science is helping solve high-profile crimes in Australia and around the world. My first question is, as a little girl, did you ever grow up thinking, I really want to be an expert in bodily fluids and DNA?
No. No, that was not my intention. But I do think I was one of a lot of young women who were highly influenced by what is now known as the Scully effect. And if anyone's ever watched The X-Files, that she was an amazing, brilliant woman and an amazing character that lots of, I think, young girls inspired to be like. She worked for the FBI and she was a brilliant pathologist, along with a broad science field that she kind of covered, not necessarily true to life. So I think she was probably a true inspiration. And I did have an interest in forensics from an early age. And I have to say that was back when you talked about forensic science and people go, oh, what's that? Very different world to what it is now where everyone knows what that means. So I think those kind of, particularly that inspiration put me into a science field. And then going through university, I fell into the forensic field as well.
It's quite funny how you say you fell into the forensic world. What were you doing at university and how did you manage to fall into forensics?
So I studied biomedical science at the University of Technology in Sydney. So obviously had an interest in that kind of medical field, a bit of biology, fluid, that kind of stuff there, but a bit more medically inclined. And then when I came to doing my honours degree, there was a project available. I had an interest in forensics and I spoke to one of my professors and said, anything going in a forensic field that might be of interest? And they said, I think I know about something with the Australian Federal Police. And so it wasn't actually a published project at the time, but they contacted them. And so I went and had a visit with them and interviewed for this project. And they gave it to me. I was super lucky. I don't know whether anyone else even applied or anything, but I showed some interest. And so I then was offered to do this project for the Australian Federal Police, but it did involve me moving from Sydney, where I was living, to Canberra to do this for them. And I kind of fell into it because I did this project for them and they were such an amazing organisation to work for at that particular time. They gave me a little job as a student in there and I would retrieve case files, I'd shred paper. Nothing very interesting, but it was a great little job that I could do on the side of my honours project. And at the completion of that, they offered me a position to go into their reporters training program. So that was a training to be a reporting scientist. And that was in their biology field. So I was doing a forensic DNA project. And then they offered me to become a reporting scientist in forensic biology and DNA.
What is a reporting scientist as opposed to an investigative?
So a reporting scientist is somebody who is sufficiently trained to be able to look at the data and the results of testing. They're not necessarily doing all the testing themselves. They may do some of it. And they then will go to the courtroom and they'll report the results to the court.
So do they actually analyse
depending on the laboratory that they work in, some of them will do. And working in the biology team in Canberra with the AFP, that was quite a small team at the time, that you would examine your cases yourself. You would collect samples for DNA. You may not necessarily be doing the DNA testing, but you have to be experienced in how to do those tests. And I've spent a lot of time doing those DNA tests in the lab, processing those DNA samples. And then you interpret the results and you take those results and then you report them to the courts.
Are you attending any crime scenes there, or are you predominantly lab-based?
No, again, that's dependent on the jurisdiction. So in Canberra, we were scene attending for your more serious scenes. So you wouldn't be a routine crime scene examiner, but if crime scene needed some specialised support, they would call you out to a job. Whereas in jurisdictions like New South Wales, the forensic biologists there are not scene attending. They fall under the Department of Health.
Not the state police?
Yeah, so they're not part of police.
Ah, that's interesting.
And what happens now within New South Wales is the laboratory, known as the New South Wales Forensic and Analytical Science Service, is not even necessarily examining that many items themselves anymore. Police collect a lot of their samples. They put them in what they call a robot accessible tube. It goes into the laboratory and they're just processing those samples for DNA. So it is then the police people that are attending the scenes and collecting the DNA samples and examining the items.
When you are therefore examining in the lab and taking extracts of the DNA samples, do you have any idea about the story behind that sample or where it's from? How much information do you actually have about that sample if you're not taking it yourself?
So it's a real challenge, I think, because I've worked very briefly at the New South Wales Forensic Analytical Science Service when this process was coming into place. And I think it's quite a challenge because all they're really doing is putting a sample into their robotic line and reporting a DNA profile. There is a bit of a disconnect between the biological examination and any biological fluid testing that's being done and the reporting of the DNA. That is now separated. So when it gets reported to the courts, they get the results of the DNA, and then there'll be a separate report that gives them the results of the biological fluid testing.
Is that a good thing or a negative thing? Because I can think of pros and cons for both.
It's very much a personal opinion. Personally, I don't think it's the best system. And I think what they're doing is siloing the information. And I think it's very easy for things to get lost in the process when you've got these silos of information that then is trying to come together at the court.
Can you explain that a little bit more?
For example, all it takes is for, say, the crime scenes examiner statement to be read out on the court and evidence presented. And then it's not going to happen back to back, but down the track, then the DNA results get presented in court. I feel that sometimes there's going to be a disconnect between the results of that testing and that particular result being presented in court in terms of the identification and the DNA. So they're not necessarily going to recognize, oh, that's from that blood stain that was collected on that clothing here that now matches that person. So the lawyers need to be very careful about how they're presenting that evidence to ensure that they're giving the full picture to the courtroom. So I think it becomes more challenging for them in the courtroom to make sure they're generating that complete picture of the evidence.
Is that like putting it in a context? I'm just thinking, if there's a family using the same washing machine and their clothes are washed together, will the clothing actually get each other's DNA on it from the washing process?
Yes, that's a possibility. And that's what one of these fields that's really exploded in the field of forensic DNA of late, because the DNA testing has become extraordinarily sensitive. You know, they can start to identify DNA information from as little as 6 cells. When you're thinking an individual shedding hundreds of thousands of cells a day, and a lot of these will be on the clothes that we're wearing that then go into the washing machine, then gets all mixed up. But it's also very dependent on a number of variables, such as the cycle that you're using on your washing machine. Are you using hot water? Are you cold water? Are you pre-treating? Do you have enzymatic washing powders? There's a huge number of variables. But what we do know is that, yes, it is a real possibility that when you commingle clothing from different people in the washing machine, that you can transfer DNA. You can also transfer sperm cells and other biological material.
If you, for example, have somebody accused of sexual assault of a family member and you look at underwear and there's evidence of DNA inside someone's underwear. If you just take that to the lab and you as a scientist have no context, you can report there's male DNA on there and there's female DNA on there. Who then does the comparison with the alleged victim and the alleged suspect?
So that would be done by the biology laboratory in terms of the reference sample comparisons. The Forensic Biology Laboratory, New South Wales FAS, would be doing that. It's in New South Wales. Queensland's in the Department of Health, Victoria's within the police force. But it's that forensic biology unit, whoever that is, that will be doing the comparison to the reference DNA samples. There would be also the individuals that would be uploading any unknown profiles and crime profiles onto the National DNA database.
So as the scientist, you don't have that information that these people may have shared a washing machine?
Not necessarily. So what also you find here is that some of these reports can be quite factual, and they're not necessarily disclosing some of those limitations to the evidence. That would be something that would be drawn out in the courtroom when the scientist is giving evidence. So again, you need the lawyers involved to be very aware of the issues around the evidence and the limitations to the evidence, so they can help disclose them to the evaluators of the court so they can evaluate this evidence appropriately.
I can understand why you're not given that information to remove bias, and motivational bias is something you talk about. But there is also a lack of context bias.
There is.
So what is the solution to giving better outcomes? Because you can't expect lawyers to understand everything about forensic science and the absolute intricacies and sensitivities and false positives, false negatives. Lawyers aren't scientifically trained generally, nor are judges.
No.
So what is the solution? Because you clearly need to eliminate bias of the sample, of the person taking the sample. But you also need a context to make that evidence make any sense whatsoever.
Absolutely you do. And to be able to appropriately evaluate the evidence, you do need to know things about the origin of the sample, what it's been tested for, how it's been tested, because each of the tests that are used have their own set of limitations as well. Then you need to look at the DNA information and who that's matching to, and the mixture. If there's mixed DNA profiles, they're DNA profiles that originate from more than one individual. So there's a lot of information that can be obtained in relation to a particular sample. And what we talk about in terms of risk mitigation, in terms of your bias, is what we call a sequential unmasking. So that is performing your tests and then looking at the actual contextual information for that particular case, and then performing your more tests, and then performing your evaluation and then getting the information. So you're not being biased in your interpretation, but you're then getting in the information to provide you the context.
Because presumably people who handle money, credit cards, I'm thinking people who work in retail, taxi drivers, they're handling someone's credit card, for example, more frequently than anyone else might. And so if their DNA is found on a credit card, for example, where there's a crime and the credit card's been recovered, how do you put that into a context when you're just finding multiple DNA on an item?
So that can become quite challenging. And I think we're in very much a world now where our DNA testing is so sensitive when it comes to this, what we call trace DNA. So this is DNA that we can't attribute to a biological fluid. So it may be due to the nature of the sample, that it's just a swab from a door handle or a swab of a credit card, something like that. There's been no biological testing. Or we've got maybe some biological testing, but we've got mixed DNA profiles, therefore we can't attribute that biological testing to an individual. We call it trace DNA. Its biological origin is unknown. We're in a world now where our DNA testing is so sensitive that it's almost better used for intelligence purposes as opposed to having strong evidentiary value, because we know that not only can people deposit DNA onto an item directly because they've physically handled it, they can also deposit DNA onto an item in what we call in an indirect manner. So that means that DNA has been deposited through some other means. And so that can be transferred by another person or via another object, or just by being in a particular environment. Like I said before, individuals shed hundreds of thousands of skin cells in a day. So you can walk into a room and be shedding your DNA, even if you haven't particularly touched anything. You can also do it by speaking. You can spread your DNA up to meters in front of you just by speaking. And it only takes about 30 seconds of speaking in an environment for your enough DNA to be transmitted that you could be detected. They're also talking about what they call eDNA now, where it's environmental DNA, that DNA can be transferred through air conditioning systems and your pets can transfer your DNA and all of these kind of things. So necessarily having DNA that can't be attributed to a biological origin such as blood or semen or saliva in an environment really isn't going to have strong evidentiary value, because there's any number of ways that DNA could have gotten there.
So has DNA actually become so sensitive and so well refined that it's done itself out of being effective evidence?
In some respects it has. And I think we're going to see more and more where the courts are going to make applications to what they call the evidentiary value is actually more prejudicial to the defendant than it is actually probative. It's not giving you good evidentiary information, but it is actually giving you prejudicial value against the defendant, because juries still put a lot of weight onto DNA. And so they hear that there's a DNA match, and they do think that that person must have been there. And I think what scientists have not been doing a great job at of late is conveying the limitations to that particular type of evidence, in that it really does have strong limitations around it. You do need to interpret it with caution. And yes, it may be present within a crime scene, but it's not evidence that that particular individual was actually in that crime scene or within that crime scene at the time that the crime event happened. There's a whole background to the scene, and we don't know that history. Have they had reason to have contact with that environment in the past, or individuals that have been there in the past? Is there something that bears that someone in that house has had contact with? There's a huge number of variables that we don't know when it comes to the history of a particular DNA profile getting into a scene.
Well, I think just to sort of get my head around it, it used to be when there was a hotel room, there was a crime in a hotel room, police would be just, what are we going to do? There's going to be DNA everywhere from how many people have been in this room before. And through this podcast, we've found out that 10 to 25% of the DNA on your hands is someone else's. So with those two things in mind, I'm now thinking that a lot of scenes will be like a hotel room, even though they're not a hotel room, because there will have been that much transfer indirectly and directly of DNA. So I would call it a false positive if someone's DNA is found at a crime scene they never were, but it's probably not the correct terminology. What would you call that?
No, so we just talk about them being transfer events. So their DNA has been transferred into the scene by some manner. But what we're finding here is that it's almost a reversal of the onus of proof. So the prosecution is supposed to prove their case beyond a reasonable doubt. But when it comes to this trace DNA, what we're seeing now is that it's almost up to the defense to provide an explanation for how their DNA could possibly have been in that particular location or in that scene, even when it's trace DNA. So they're almost having to prove themselves to show that there's an explanation, a logical explanation for how it could have gotten there. So they're reversing that onus of proof. It's not prosecution proving that, it's defense proving that they've had some legitimate contact with the scene, or people that have been there, or, some connection to the environment by some manner. And you've got to think that this is also extraordinarily challenging to do too, because we know DNA doesn't just get transferred once. It can get transferred over and over and over and over. And we do know the amounts diminish over time, but there's also a lot of variables in terms of the nature and duration of contact that we don't know. So we don't know how much DNA is going to get transferred and potentially be available for onwards DNA transfer. So it's a very complicated and challenging field, a very difficult situation at the moment.
That sounds counterintuitive, that the onus is on the defence to prove how that DNA got there, because how do you prove you were never in a place? How do you prove you never met someone? It's extremely difficult to prove something that never happened.
Absolutely.
I would have thought that you'd be actually giving defence like an easy pass, a free pass, saying, look, DNA could have got there anyway and we don't have to explain it. Whereas I would have thought the prosecution onus was on saying how that DNA got there.
Yes. But yeah, that's not necessarily what we're seeing in the courts at the moment. Like they'll just put it out there that DNA's been located in that location or whatever it's on. And so it may be that, I don't know, there's a swab of a door handle inside a house where a crime event has occurred. Someone's been identified on it, on that handle of the swab. It's a mixed DNA profile. They can't be excluded as being a contributor. Therefore, they've been essentially identified at being at that location. But they're not necessarily disclosing the limitations around that evidence and trace DNA unless the witness is giving evidence in the witness box and it gets drawn out in evidence. Because a lot of the times you may generate a report for the courts, but you're not physically giving evidence. I've generated lots and lots of reports for cases, but I don't actually physically give evidence in court. So it's the reports that are giving that limited view of the evidence, and it's not until you get a scientist in the witness box and you get to probe them about the limitations of the evidence that those things are disclosed.
Do you then feel a responsibility when you write a report in case the evidence can't be expanded on and its potential flaws or limitations be declared in court? How do you then structure your report?
This is a lot of what I do now, because now I don't work for a policing organisation. I'm an independent consultant, is that I really disclose these limitations up front and talk about the limitations to trace DNA and how it can be transferred and all of those types of things about direct and indirect DNA transfer before I actually talk about the results. So to go, here's the information about trace DNA, here's your result. Remember, this is a trace DNA sample, therefore it's subject to the limitations. So that the court, even if I'm not giving evidence in court in this particular matter, that they should have a greater understanding about what that evidence means.
We mentioned motivational bias. Can you give us a couple of examples of how motivational bias works and the effect it can have?
Motivational bias, as much as people that work within law enforcement and some of the agents that test forensic evidence for law enforcement agencies, would like to pretend that they're not biased. But unfortunately, just to the nature of the work, the instructions that they're receiving, they do naturally have an inherent bias in the work that they're doing. I've had one report from an examiner within a law enforcement agency that said that they'd screened an item with light source. So there's an allegation of an oral sexual assault. So, detection of saliva was probably pretty important. Their report literally said they screened the item with an alternate light source. So that's a light source that gives a wavelength of light other than white light. It's not a torch, it's a coloured light. Your nice, fancy blue CSI lights that people like, but they also come in different colours or different wavelengths of light. So they screened this item with this torch. There was some fluorescence. And we do know biological fluids such as saliva can fluoresce. Semen can also fluoresce. Urine can also fluoresce. Some cleaning fluids and washing detergents can also fluoresce. But their report said, I screened the underwear with an alternate light source and fluorescence was detected, indicating the presence of saliva. They collected a sample from that, submitted it to the laboratory. So the report went with, they had a test, if you call it a test, indicating the presence of saliva. And then the DNA test went into the laboratory. But that is an incredibly misleading report, and it's biased because they're saying that there's an oral assault. They had information that was an oral assault. They've then assumed that it's saliva, even though it could be a number of other biological fluids, or it could have been cleaning, a cleaning fluid or, washing powder or washing detergent. And that's gone into the court when with nothing about the limitations of that evidence. And the thing about that one that particularly irks me is that an alternate light source isn't even a test for a biological fluid. It's what we call a screening tool. All it is to help you find substances that warrant further testing, because we know that they will react with many other substances other than one particular biological fluid. Like it could have been even one of another, it could have been 3 biological fluids that fluoresce like that. let alone the other substances that are not biological fluids. But they've been biased by the information. They've detected saliva with their light, and they've written a report as such.
And they've spoken in absolutes, which is always a concern.
Yeah, absolutely. That's right.
Had they said consistent with, is that still misleading?
I think it is, because they're not actually doing a test for the presence of saliva. All they're using is a tool to help you locate it. So what they needed to do was to screen. I think, yes, they found fluorescence. They needed to go on and do some further testing to then actually try and narrow down what that biological fluid is, which they would have had those tests available, but they didn't do.
That's so interesting, because in all the sort of television and film and stories, we have experts being examined and the defence lawyer is always saying, is it possible? Could it be something else? And the moment they say, yes, it's possible, and they don't give a percentage, like it's 1% possible as opposed to 99% probable, does this mean that now expert testimony is going to be sort of more wishy-washy and less helpful in prosecutions and in defence?
Well, I think it's always been the case when it comes to, the scenarios that are put forward to that it's very difficult to evaluate the probabilities of things. And so I'm a biologist, so I work a lot with numbers. We like maths. That's what biologists do. And that's what sets us apart from some other forensic disciplines, is that we work pretty much in numbers. And what we would like to be doing if we're evaluating this evidence is to put it into what we call a Bayesian type framework. And this is where we have data about what will produce, say, if you use a sexual assault, for example, and you have spermatozoa located within the vaginal cavity, we can say that, so if the sexual assault kit is collected, say, 3 days after the alleged offence, we should have data that looks at how much sperm we would expect after 3 days from a post-ejaculation event. And then you could put your numbers into this evaluative framework and give you a probability for the evidence that it is this many times more likely that sexual assault, the ejaculation or the deposition of sperm into the vaginal cavity occurred within 3 days, as opposed to it being a transfer from, say, or for it not there being no ejaculation into the vagina and the DNA has been deposited through sampling methods or something else. So we like to work in those kind of numbered frameworks where we can. And that's where we're not going to be able to do that to generate probabilities for evidence like the screening tool. Is it saliva or is it something else? What they really needed to do is do more biological testing to narrow down the origin of that biological fluid.
So the only way to mitigate this is to do as much biological testing on every sample, but clearly that's not feasible financially, time-wise, collection-wise. Is this a solvable problem?
No. So it's always, and I don't think there is a great answer out there given the technology that we currently have. There has always been this balance between doing your biological testing, because you have to remember testing for a biological fluid is consuming material. And so testing your screening test, or what we sometimes call a presumptive test, doesn't generally take a huge amount of material. You can wipe a tiny point of a filter paper and collect a sample and do your test on that little filter paper without removing too much. But things to go on and do your confirmatory type testing that are going to give you more definitive answers about the origin of a biological fluid require more material. And sometimes there is that balance of being able to conduct a confirmatory test, or do you need to conserve that small amount of material you have for your DNA testing and your identification purposes? And more often than not, you have to sacrifice your confirmation testing or your confirmatory testing for your DNA analysis, the identifiable information, because that's what's going to give you more information. But there are tests out there that have been around for a little while that are still not quite right, but it's where you'll be able to do your biological fluid within the DNA testing process. Some of it's known as what we call mRNA analysis. That's your messenger RNA. And so there are some tests where you can do that within your tube of your DNA sample, and you can then work out what biological fluids are actually present within your DNA sample. So that's kind of going to be your best solution when those kind of tests become more widely available and more robust.
Because messenger RNA is in how many cells? Where is it?
Oh, so it's in a lot of places. So it's freely available. It's much more abundant, within our bodies. So we'll be able to do those kind of tests, and you're not then compromising the DNA that we're trying to target as well. So they're two different things that you're targeting.
So now with the limitation of DNA, we're moving into messenger RNA. And if we get more sensitive with that, that can give us a lot more information.
Absolutely. So, and it's about targeting these regions, which they've already got. ESR in New Zealand were great pioneers of this mRNA testing, and looking at, doing your definitive confirmation tests effectively, because it's based on your RNA of what's within your DNA sample. But it's narrowing down those parts of the RNA that give us those really robust, this is that particular biological fluid. And they were doing so well with this that they were even being able to differentiate between just blood and, for example, menstrual blood, because sometimes that can be an important question that requires answering. So, it's about getting those targeted areas and getting your targets right to be able to do that testing, and getting it really robust. And I think where there's strides in this development field, because that's going to really advance the forensic biology, and it's going to help mitigate some of the issues and challenges that we see with forensic biology evidence at the moment.
In terms of data collection now, there's a whole database, there's a whole lot more data that's been collected. Are you using algorithms and artificial intelligence to help identify DNA samples and relate them to suspects or people already in the DNA database?
There's kind of two aspects to this. So there's our routine testing that's performed. That's where we use our STR kind of markers using autosomal DNA that comes from your mother and your father. That's the routine testing that's performed. And those DNA profiles from crime scene samples will go onto what we call the National DNA Database. NCID, it's called the National Criminal DNA Identification Database. And that's a nationwide database. So, and that cross matches against all of the jurisdictions in Australia. So crime scene samples go onto their convicted offenders. So if someone's been convicted of a serious and indictable offence, their DNA profile will go onto the DNA database and it stays there forever. Suspects, if there's a suspect and they've been compelled to give a DNA sample, they will go onto the DNA database for a period of 12 months. So generally, no, we don't upload volunteer samples usually. So that will go get cross-matched. So that's where most of your routine matching occurs, and there's searches that go against that DNA database all the time. They'll just do batch uploads and do a search of the DNA database to see if there's any hits there. But what they're doing more in terms of more advanced searching, they can do what we call a familial search, and that's where they can search the National DNA Database for non-matching DNA profiles. So that's DNA profiles that are similar but not quite the same. But they'll indicate the presence, or they could indicate a familiar relationship between individuals. And that requires a bit more investigative work, because you will get people that have components of their profile that are the same but are not actually related. But it can give investigators good leads. And then there is what's become a big kind of topic in the area of forensic biology at the moment. It's what they call the forensic investigative genetic genealogy, the FIG. And that's where you're using more specialised DNA testing, what we call sometimes SNP testing. We're searching other areas of the DNA. We're looking at single changes in the DNA strand, and then looking at extended familial searching. And so that's where, particularly in the US, they're having enormous success with that at the moment. And we're starting to get on board with that here in developing our FIG capabilities within Australia.
Well, with so much theft, AR has been known to use a lot of intellectual property and accumulate to educate itself and improve. And we've got so much more theft of intellectual property because there's so much data available. Is DNA at risk? If we've got identity theft as well, is DNA at risk of being stolen and misused? Well,
potentially, yes. And I don't think a lot of people realise, like, I don't know how many people have actually signed up to that, but lots of them that would have done an ancestry DNA test, and they've taken a sample of their DNA and they've submitted it off to the laboratory and got their lovely ancestry information. That they need to be really careful when they're filling out the forms for that, because that DNA, when you're submitting it, you're pretty much giving it away. And so I think there's tick boxes about not sharing the DNA, but it may go into their DNA databases. And there are instances of companies buying those DNA databases for information, maybe that might go on for medical research and those kind of things, or they can also be used for things like big searching, forensic investigative genetic genealogy searching. So they may inadvertently even be then used to assist police in solving crimes and things, even if you didn't necessarily sign up and give permission for your DNA to be used for that particular purpose. So I think within Australia, we need to be working on some legislation around, if you're giving your DNA to organisations like this, about what your privacy, what limitations there are, and what you're signing your DNA away to be given to, whether they are allowed to be used for, say, police searching, or what you're going to give them permission to use your DNA for.
And if they are going to do familial searching, could you actually be implicating a relative who's committed a crime?
Potentially.
That may be a motivation to do it. Who knows?
It may be, absolutely. Or it could be an unwelcome surprise.
Coming up, a chance discovery plays a part in helping police solve one of Australia's most renowned murder investigations. There is actually, it's a notorious case for all the wrong reasons that happened in Sydney, and it was called the Lin Family Murders, and it happened in Epping in Sydney. A nice suburban newsagent's owner's family were murdered. And I gather you were involved in actually collecting a sample there that has since been deemed all through the media and the courts as stain 91. Can you please talk us through what you knew about the case when you were there at the scene and collecting stain 91?
I actually had a long involvement with that particular incident. So I was involved right from the initial investigation when the family were discovered in their house, and so had done a lot of examination and testing of samples from that particular scene within the New South Wales Police Laboratory, because I was working for New South Wales Police at the time when New South Wales Police had their own specialised biology unit. That unit no longer exists, but I was there as a specialised biologist. So we were scene going, and we also did laboratory examinations, and we also did laboratory examinations in conjunction with other disciplines. So like with fingerprints and documents, that we could work together to maximise the forensic evidence on items. So I have a long history of working on that case, a lot of long hours and late nights, and, you know, it was very obviously high profile and serious matter, and obviously there was great concern in the community because of the seriousness of that offence.
Just to explain to someone who doesn't know about the Lin family murders, what did they involve?
It's a very tragic incident where a family of 5 were actually found deceased in their house, and they were essentially beaten to death. It was a very brutal kind of scene, and it was a very confronting and brutal scene, and it was very high profile that a family, like 5 murders in one night in one family to have occurred, and there was no known offender at the time. Obviously, the police investigation goes on and they're trying to identify that offender, but it started to drag on where there was no one that was arrested quite quickly in relation to that particular offence. So it was always considered quite a high profile and serious case, and it was very unnerving for the community at the time. It's one of the most probably brutal examinations and cases that I've worked on because of the number of deceased and the way that they were killed. So when it came to the prolonged investigation of loads and loads of items coming from the house and looking at the bloodstain pattern on some of these items, and can you find foreign bloodstain? Is the offender bleeding? Can we find any other individuals when we're sampling these particular items? So we did a lot of items for that particular house that came out of that house. I actually went to then it turned out to be a neighbouring house to where Stain 91 was located, because a person had phoned in and said, oh my gosh, I think there's blood on my fence. So I went out and examined with a colleague of mine. We went out and examined this area where they thought blood had been found on their fence. And it turned out it wasn't blood at all, but they have obviously heightened and scared in the community. And they're like, oh my gosh, could my house be part of a crime scene? But yeah, so we had that as well. And then it was sometime later, it's got to be a year and a half, 2 years maybe, since the initial crime occurred, that this search warrant occurred at the house next door to where we'd done the examination on the fence. And Stain 91 was located in a garage. And so myself and 2 other biologists went out to this scene because we knew there was a lot to search. We started at this garage and it was full of stuff. Like, just think about someone's garage and it's got furniture and items and ladders and tools, and it was just full. And so we spent days screening every item in that garage and removing it from the garage and making sure that there was no blood on any of the items that we took out. So after days of searching and thousands of tests, the garage is finally clean. And so we then were at the point where we started screening the floor. And so this is the way biologists do these kind of tests. It's not like crime scene do, where they might spray luminol and they're looking for big major reactions and things like that. We're looking at the details. So we're doing little spot tests on anything that looked like it could potentially be a blood stain. And so you're using your little filter paper and your chemical reagents. Is it changing color? No. Discard, like moving on. And so we finally get to this floor, and there's 3 of us working on our hands and knees, spot testing any discoloration on the floor. And I think in the end, we sampled 5 discolorations on the floor where we got reactions, positive reactions. And one of them was that infamous Stain 91, which then went on to generate a DNA profile that was a significantly mixed DNA profile. I believe it was a mixture of 5 individuals they were able to estimate that then matched the family.
Where was the stain?
On the garage floor. It's pretty much towards the middle of the garage floor.
Was it under anything or disguised or hidden?
Yeah, no. So it would have been under. We would have probably walked all over it while we were screening and taking things out of the garage. Like, there was no way that it would have been obvious, like, because when you were going in that garage initially, like, you had to kind of weasel your way in between furniture and things like that to be able to, there was no clear walkways within the garage because it was so full of stuff. And so by the time we'd cleared all of the things out of it to give us some workable space in there, we'd screened everything in there. And then, yeah, we tested the floor, and it just happened to be on the floor there.
Is there any theory as to what actually got that stain there in the first place? What did you surmise, or what can you surmise, even in retrospect?
Actually, this was obviously very important evidence to the court, right? With that, particularly the detectives and things wanted to know about how this stain got there. And what I was up to, because I also in my training have done bloodstain pattern analysis. So what I was able to tell the detectives and went into my report at courts is that it wasn't a drip of blood. So it's not a blood that's fallen under gravity and just landed as a stain on the ground. The nature of the stain is that it was like what we would call a swipe mark. So that's where a bloodstain item has made contact with a surface and left a mark on that surface. That's what it looked like. What made the mark, I don't know. And this was one of those samples too where we had to compromise, like I spoke about before, is that this stain was old. It was only a light stain. It did give us a very good reaction to what we call our presumptive test. But that presumptive test also reacts with other things, including some plant materials, rust, and other substances, not just blood. It doesn't tell us that it's blood, but we had that positive test. But we decided that we were in a better position to sample that stain for DNA testing as opposed to trying to consume material or a confirmatory test. And like I said before, those confirmatory tests require more material. So the risk is then too is that we conduct a confirmatory test after we've done our DNA sample. And because there's not enough material there, you're just not going to get a result. Well, you don't know whether it's going to be positive or negative, but did we get a negative result because there's not enough material there or because it's not blood? So we didn't actually do a confirmatory test because it wasn't going to really give us an answer.
How large was the stain that you're talking about?
From memory, it was only about a centimetre, maybe a centimetre and a half.
Wow.
In length and maybe half a centimetre wide. Like it wasn't very big, and it's obviously it was old. It wasn't like a direct deposit into the concrete, like a drop falling down where you may have had more material. It was a swipe type mark, so it's a transfer mark.
It's a very superficial, not deep, nothing.
Yeah, it's not like there was lots of material that we could have dug out with a swab, or even if it had been a drip mark, we might have even tried to chip out pieces of the concrete, but it wasn't that type of stain, unfortunately. So that's infamous stain 91. So obviously it was made a pretty big deal of in court. I think I gave evidence in the 3 trials when that occurred because there was a long court process for that particular matter as well.
Was it challenged?
Yes, of course it was, and justly and rightly so. Defence were well within their right to question about what we had done, how we'd done our particular testing, and what the type of test that we used. And it was interesting because one of the things that they did talk about is, could it be a false positive? And I'm like, well, yes, it could be. And part of what I'd seen at the house next door is there was a plant on that fence line that was also, its sap was giving false positives to this blood testing reagent as well. But when you did the test, you knew it wasn't blood that you were testing because it was sticky and it had this funny texture to it that blood doesn't have when it's dry. And I was pretty sure this was. And then I actually took a leaf from the plant, cracked it open, and then did a test. I'm like, yeah, that's where those particular positives are coming from at that scene. And he's like, well, could that be in the garage as well? And I said, well, I don't think so. And again, I wasn't talking in absolutes because it didn't have that sticky nature that had been seen in that plant sap material. It was dry. We'd also conducted, I don't know how many tests, but we went through boxes. We have boxes of filter paper that are in packs of 100. And I probably went through 5 packs of filter paper on the garage floor alone, let alone the other 2 scientists. They probably did the same. So we're talking about in the order of 1,500 tests that we've done on the garage floor. And we only had those couple of positive results that didn't have that sticky type material. I was pretty confident that it wasn't a false positive because of the plant material outside.
Was there criticism of how the family's DNA could have actually got into the garage as opposed to a blood stain any other way?
All I could say is that we've identified this material. It's tested positive to a blood test. It's given us a positive here. And to me, it looks like a swipe mark. That was pretty much the limitation of my evidence. I didn't deal with the DNA results in that particular case. That was up to the New South Wales FAST scientists who then talked about their DNA there. But it would have come down to, again, because we couldn't confirm the presence of the biological fluid, that it's trace DNA effectively in that scene. But then you've got a sample that just happens to match 5 individuals for that particular family in that particular location on the floor. That has given a positive test to a presumptive test for blood, even though we weren't able to confirm it. So I think it's putting all of those pieces of information together to try and get some clarity as to what that could have come to, and for the evaluators, the court, to decide what that actually means.
And obviously the prosecution had to establish a motive and opportunity, and they have other circumstantial evidence as well.
All of that elements of the criminal offence that they have to prove.
How important do you think stain 91 was in the case, looking back?
Yeah, given the attention I got, I think it was pretty important. I spent, a lot of time in the witness box talking about stain 91 and what it could be and what it couldn't be and how it got there. And while I was aware of a lot of the case, when you're working in these particular jobs, you don't necessarily have the whole picture of all the evidence that's been uncovered either. So I was just very focused on, this is my little element, and I don't see the rest of the case that's been built there. And because I don't necessarily need to be biased by anything else that's going on either, so you don't tend to have a huge awareness of what else has gone on in that particular matter.
The challenge for that would have been that this case was absolutely smashed by the media. It was constant coverage, and because there were retrials and it was this saga. It was almost like a soap opera saga, the way the media treated it, and yet there were real victims, and and there was one surviving family member, a daughter.
There was,
and
yeah.
she had to endure all of this as well. And a family member was the suspect.
Absolutely, he was. And it was ultimately convicted.
How do you feel about that case now? How did that affect you personally?
And to me, it's just one of those long matters that you have involvement for such a long period of time, from when the incident first happened to the completion of my involvement was, it's got to be 5 years or something. It's a long period of time. So you do live with those cases. But I think when you work in this kind of job, you just need to be very good about separating your work from home. And, you know, you're also dealing with other cases as well. So you can't have all your attention focused on this particular matter. While it's important, you can't do a disservice to the other aspects of your job and the other cases that you have to work on as well. So it's a balancing act, really.
Looking back, how did you handle the media attention and the profile that suddenly you were thrown into the world having?
I didn't really have personally a lot of media attention on me, apart from, like I said, there was media at that job that I went to examine the fence, but there was no media when we were doing the search warrant on the person of interest house. There was no one there for that. And even giving evidence at court, there was no media attention on me either. You do try to keep a low profile. You don't talk to anyone when you're going into the courtroom, and you don't talk to anyone going out, and you're very cautious about anyone that might approach you. Not that I actually did, but that's part of your court training, is you don't talk to anyone about anything. You know, you don't talk to people, general chit-chat going into the courtroom or anything, unless they're people that you're going to see. So really, I didn't have I wasn't impacted by the media at all directly. And then you just screen yourself. Like, I didn't read any of the news articles or anything, even though it's on, you know, you can't really escape it. You're turning off the news if it comes on and those kind of things. You're just screening yourself from the chaos essentially of it. So you talk about there being so much media and about it, the kind of furor that went around that case. And I know that was the case, but I don't know the details of what was being said or how that was really portrayed in the media.
Jae, thank you so much for sharing your expertise. And I think it's incredible that DNA has come so far. It's now questionable, and it's now debatable and controversial at times. But let's hope that the next step, the messenger RNA and future developments, will take us to a better place in terms of evidence in crimes.
Well, thank you so much for having me. And I hope you got as much out of the chat as I did. I thought it was lovely.
Crime Insiders Forensics is a LiSTNR original production. It's hosted by me, Kathryn Fox. Production team includes Holly Mitchell, Link Kelly and Jade Murray. Executive producer is Dan Mullins. That's all we have time for this week. As you can probably imagine, I'm a big fan of all things crime. I discovered a fantastic podcast on the other side of the world, and I'm totally hooked. It's called Red Rum True Crime, hosted by Grace Cordell. Each episode focuses on a new story. Real life, real victims, real crime. A case I'd never heard of is that of the candy man Dean Corll. Imagine trusting the local candy store owner only to discover he's one of the most sadistic serial killers in history. Dean Corll, who they called the Candy Man, lured innocent teens into a nightmare beyond belief. What drove him to commit some of the most chilling murders in Texas history is pretty unbelievable. You won't believe how deep this darkness goes. Check it out. Red Rum True Crime. You'll love it.
Hi, I'm Grace, host of Red Rum True Crime podcast. These cases focus on the true victims of crime. Why not jump in at episode 114, the tragic murder of Jasmine and Aaliyah? The main suspect in this case gave an extremely bizarre interview to a number of press reporters whilst he was drunk and reportedly high. He speaks about an awful lot on camera and has this completely inappropriate laughing and chuckling response when talking about the case. He may even have thought he was gonna get away with the double murder he'd been accused of, but what he didn't know was that 2 undercover officers were on their way to catch him out, and he easily and willingly took the bait. You can find us wherever you get your podcasts. Just search Red Rum True Crime. That's Red Rum, murder backwards, REDRUM True Crime.